Hello students, welcome to Read Med Prep Academy channel. Today in Molecular Genetics part 11, we are going to discuss DNA fingerprinting technique. The fingerprinting technique was first developed by Alex Jeffreys in 1985. He was the recipient of the Royal Society's medal in 2014. Each of us have the same chemical structure of the DNA, but there are millions of differences in the DNA sequence of base pairs. This makes the uniqueness among us so that each of us except identical twins is different from each other genetically. The DNA of a person and the fingerprints are unique. Here you can see the number of chromosomes, which are 23 pairs of human chromosome with 1.5 million pairs of genes. Out of the 23 pairs, 22 pairs are autosomes and 1 pair is the sex chromosome. In males it is XY and it is females in XX. It is a well-known fact that genes are segments of the DNA which differ in the sequence of their nucleotides. Here you can see that the cell consists of the nucleus consists of the chromosomes. The chromosomes is made up of packed DNA molecule, and the DNA consists of portions of bases which are called genes. And these genes translate the proteins to the messenger RNA. Not all segments of the DNA code for proteins. Some DNA segments have a regulatory function, while others are intervening sequences called introns, and still others are repeated DNA sequences. Here you can see the DNA consists of the introns and the extrons in the gene, and this is transcribed as the pre-messenger RNA transcription. In a mature messenger RNA, the introns are removed by splicing through spliceosomes. In DNA fingerprinting, short repetitive nucleotide sequences are specific for a person. These nucleotide sequences are called variable number tandem repeats, VNTR. VNTRs of two persons generally show variations and are useful as genetic markers. DNA fingerprinting involves identifying differences in some specific regions in the DNA sequence called repetitive DNA, because in these sequences a small stretch of DNA is repeated many times. These repetitive DNA are separated from the bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak and other small peaks, and these are referred as satellite DNA. Here in the chromosome you can see that the satellite DNA is present on either side in the telomere and in the center in the centromere, whereas the repetitive DNA is present between the telomere and the centromere. What are the differences between the repetitive DNA and the satellite DNA? Repetitive DNA refers to the sequence of the DNA in which a small stretch of DNA is repeated many times, whereas satellite DNA refers to those repetitive DNA sequences which do not code for proteins but form a large part of the human genome. Repetitive DNA is not classified further, whereas satellite DNA can be classified as microsatellites, mini-satellites, etc. Repetitive DNA does not show high degrees of polymorphism, whereas satellite DNA shows high degree of polymorphism. Depending upon the base composition of A:T rich or G:C rich, and based on the length of the segment and the number of repetitive units, the satellite DNA is classified into many subcategories, as already mentioned, the microsatellites and the mini-satellites, etc. Now, what are the differences between the microsatellite and the mini-satellite? In microsatellite, short tandem repeats that consist of 1 to 9 base pairs monomer repeating sequences. In mini-satellite, tandem repeats with a monomer repeat length of 10 to 100 base pairs. Microsatellite is rich with A and T bases, whereas mini-satellite is rich with G and C bases. Microsatellite is also known as simple sequence repeats, SSR, or short tandem repeats, STR, whereas mini-satellite is known as variable number tandem repeats, VNTR. Here you can see the mini-satellite, the variable number tandem repeats, the repeat unit size is hundreds of base pairs and it is repeated 4 times. In a microsatellite, which is otherwise called short tandem repeats or simple sequence repeats, the repeat unit size is 2 to 6 base pairs. It is repeated from 8 times to sometimes 20 times. These sequences of satellites in the DNA do not code for any proteins, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of fingerprinting. Here you can see the schematic representation of DNA fingerprinting. Few representative chromosomes have been shown to contain the different copy of VNTR. Number 1 is the parental chromosome and number 2 is the maternal chromosome, where the chromosome number 7 has been shown, chromosome number 2 has been shown, chromosome number 16 has been shown. These paternal and maternal chromosomes with VNTR compared with the child's VNTR helps to solve disputes of paternity. Below you can see the VNTRs in the chromosome number 7, chromosome number 2 and chromosome number 16. These of two individuals, individual A and individual B, the C is taken from the scene of the crime and it is compared with the individual A and individual B. We come to know that the DNA that has been taken from the crime site goes along with the individual B. DNA isolated from blood, hair, skin cells or other genetic evidences left at the scene of crime can be compared through VNTR patterns with the DNA of a criminal suspect to determine guilt or innocence. Here you can see the DNA taken from the crime scene, and this is compared with the suspect 1, suspect 2, suspect 3. And the DNA of the crime scene goes along with the suspect 2. This helps in solving the crimes. VNTR patterns are also useful in establishing the identity of a homicide victim, either from DNA found as evidence or from the body itself. Here you can see the victim's DNA, which is taken from the victim and is compared with the DNA taken from the crime scene. And this goes along with the DNA of the crime scene. This is also compared with the suspect's DNA, suspect 1 and suspect 2. The DNA of crime scene 1, if you observe properly, you can see that it goes along with the DNA of the suspect 2. So this also helps in identifying the victim as well as the innocence or guilt of the suspect. What are the steps involved in the DNA fingerprinting technique? Number 1 is extraction of DNA. Number 2 is PCR or polymerase chain reaction. Number 3 is fragmenting DNA. Separation of DNA by electrophoresis is step 4. Step 5 is denaturing DNA. Step 6 is blotting. Step 7 is using probes to identify specific DNA. Step 8 is hybridization with probe. Step 9 is exposure on film to make a genetic or DNA fingerprint. Let us discuss one by one. In extraction of DNA, which is step 1, the process of DNA fingerprinting starts with obtaining a sample of the DNA from the site of crime, like the blood, semen, vaginal fluids, hair roots, teeth, bones, etc. How this DNA is purified? This DNA is taken, the tissue dissociation is done, that is taken from the scene of crime, and the cell lysis is made, and the DNA binding is done, and again it is washed and a pure DNA is obtained. Step 2 is polymerase chain reaction. In many situations, there is only a small amount of DNA obtained from the site of crime. So if needed, many copies of the DNA can be produced by a technique called polymerase chain reaction. This helps in the amplification of DNA. Here you can see, if there is a small amount of DNA available for DNA fingerprinting, augment the amount of DNA using a technique called PCR. PCR is doing DNA replication in a test tube, where multiple copies of the DNA can be made. maybe 2, 4, 6, 8, 10, even lakhs and lakhs of copies can be prepared by this polymerase chain reaction. Step 3 is fragmenting DNA. DNA is treated with restriction enzymes, which cuts the DNA into smaller fragments at specific sites. Here you can see that the DNA is cut into fragments using the restriction enzymes. Each restriction enzyme cuts the DNA at a specific base sequence. The sections of the DNA that are cut are called restriction fragments. This yields thousands of restriction fragments of all different sizes because the base sequences being cut may be far apart, called the long fragment, or close together, called the short fragment. Step 4 is the separation of DNA by electrophoresis. During electrophoresis in an agarose gel, the DNA fragments are separated into bands of different sizes. The bands of separated DNA are sieved out of the gel using a nylon membrane, which is treated with chemicals that allow for it to break the hydrogen bonds of the DNA, so there are single strands formed. Here you can see the procedure. The fragments are separated on the basis of the size using a process called gel electrophoresis. The DNA fragments are injected into wells, and an electric current is applied along the gel, causing the negatively charged fragments to move. The gel is made of agarose. Agarose is a jelly-like substance made out of seaweed that allows the fragments of DNA to pass through. Now, the step 5 is denaturing DNA. The DNA on gels is denatured by using alkaline chemicals or by heating. Here you can see the restriction fragments present in the gel are denatured with alkali. This causes the double-stranded to become single-stranded. DNA is then neutralized with sodium chloride NaCl to prevent rehybridization before adding the probe. You can see the sponge kept in the salt solution, above which is the gel, and a filter is placed. Above that are paper towels placed. This is the procedure that is followed to denature DNA. Now, step 6 is blotting. The DNA band pattern in the gel is transferred to a thin nylon membrane placed over the size fractioned DNA strand by a technique called Southern blotting. Southern blotting is a procedure allowing the DNA in the electrophoresis gel to be transferred to nitrocellulose membrane. Here you can see the transfer buffer is taken, a platform is placed, a saran wrap is placed on the platform, and a Wattman paper is placed, above which the gel containing the DNA is placed, and a membrane is placed on the gel. Wattman paper stack is again placed, on which the paper towel stack is placed, on which the weight is applied. This helps in the transfer of the DNA from the electrophoresis gel to the nitrocellulose membrane. Here you can see how the procedure is followed, and the resultant nitrocellulose filter paper that is obtained shows bands of DNA. Step 7 is using probes to identify the specific DNA. A radioactive probe, DNA labeled with the radioactive substance, is added to the DNA bands. The probe attaches by base pairing to those restriction fragments that are complementary to its sequence. The probes can be prepared by using either fluorescent substance or radioactive isotopes. Step 8 is hybridization with the probe. After the probe hybridizes and the excess probe is washed off, and a photographic film is placed on the membrane containing DNA hybrids. Here you can see a nylon membrane is incubated with the radioactive probes. Mini satellites DNA tagged with radioactive phosphorus, and the gel is separated from the filter paper, and the filter nitrocellulose membrane that contains the DNA are placed in seal a meal bag, and then the probe is removed, and then the probe is hybridized to complementary sequence. You can see here the hybridization with the labeled probes helps in the detection of bands. Step 9 is the exposure on the film to make a genetic DNA fingerprint. The radioactive label exposes the film to perform an image of bands corresponding to specific DNA bands. The thick band and the thin dark bands are obtained on the film, a pattern of bars which constitute a genetic fingerprint. So what is a DNA fingerprint? It is a pattern of bands made from specific DNA fragments of an individual's DNA. To sum up the entire procedure, let us discuss with this image. The blood sample is taken which contains the DNA. DNA is extracted from the blood cells. DNA is cut into fragments by a restriction enzyme. The DNA fragments are separated into bands during electrophoresis on an agarose gel. The DNA band pattern in the gel is transferred to a nylon membrane by a technique known as the southern blotting. The DNA probe binds to specific DNA sequences on the membrane. Excess DNA probe is washed off. At this stage, the radioactivity probe is bound to the DNA pattern on the membrane. X-ray film is placed next to the membrane to detect the radioactive pattern. And the X-ray film is developed to make visible the pattern of the bands, which is known as a DNA fingerprint. What are the applications of DNA fingerprinting? One is forensic analysis. It can be used in identification of a person involved in criminal activities, for settling paternity disputes or maternity disputes, and in determining the relationships for immigration purpose. Here you can see the identification of the criminal suspect. The DNA taken from the crime scene matches with the suspect 2. There are 3 suspects and the crime scene DNA matches with the suspect 2. In paternity issues, the DNA of the mother, the DNA of the child is matched with the DNA of the father if there are any paternity issues, or the DNA of the child and the DNA of the father is matched with the DNA of the mother. Number 2 is pedigree analysis. Inheritance pattern of genes through generations and for detecting inherited diseases. Here you can see the sickle cell pedigree, where the parents, how they transmit the disease to the children. The male child, which is designated as a colored box, is a sufferer. The circle with the half color is the carrier, female, and the normal, female. You can see how the pedigree can be detected by the DNA fingerprinting. Number 3 is conservation of wildlife. Protection of endangered species by maintaining DNA records for identification of tissues of the dead endangered organisms. DNA forensics is used in wildlife conservation as well. How? An example I will tell you. African elephants are highly vulnerable species. Poachers slaughter the animals mainly for their precious tusks, have reduced their population dramatically. So to stop the ivory trade, African officials now use DNA fingerprinting to identify the herds from which the black market ivory has been taken. It is useful in also anthropological studies to determine the origin and migration of human populations and genetic diversities. Scientists are using DNA typing to help piece together thousands of fragments gathered from the Dead Sea Scrolls. DNA typing can determine the degree of relatedness among human fossils from different geographic locations and geologic areas and eras. The results shed light on the history of human evolution. Scientists have used DNA fingerprinting to identify the remains of Tsar Nicholas Romanov II of Russia and his family, executed by the Bolsheviks in 1918. They compared the DNA from the bones with DNA from the blood samples of the living descendants of Nicholas II. The results of DNA typing disproved one woman's claim that she was the Russian Grand Duchess Anastasia and had survived the Romanov massacre. So, anthropology has been useful in solving many historical murders and many comparisons with the previous eras. Today, in Molecular Genetics Part 11, we have discussed the DNA fingerprinting technique and its application. Thank you. Kindly subscribe, like, share and comment the channel Read MedPrep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.